Thank you for the introduction. This presentation has been titled Genetic Basis of Cecum Metabolome Composition and Heterogeneous Store Rats Identifies ABC and SLC transporters and enzymes including CES, CYPs, and UGTs as associated with metabolome abundance. Introduction: all metabolites in the small intestine are influenced by a number of factors. When we talk about metabolome, it can be identified as a collection of low molecular weight molecules found in the intestine. These metabolites are going to be influenced by ingested exposome, basically diet, host secretions, and also microbial transformations. We expect that at least the last two factors are going to be partially influenced by host genetic background. The objectives of this specific project was threefold. First, to identify the associated regions for cecum metabolome abundance and a heterogeneous store rat population, to estimate SNP-based heritability for those specific phenotypes, and to suggest a potential mechanisms driving a metabolite abundance in cecum. Materials and methods. This is the whole graphical abstract for this specific project, but I'm going to focus this talk on the metabolite quantification and gene mapping for these specific traits. We use the heterogeneous store rat population you already are familiar with. This is a mosaic, a genomic mosaic coming from a total of 8 different founder strains. We use a total of 1,146 heterogeneous store rats, including both sexes, and those individuals were held at 2 different centers, Michigan and Buffalo. Those animals were genotyped for a total of 5.4 million markers using the RN7 assembly as a reference. And the association analysis was performed using GCTA loco. The association threshold was 5.8, and the SNP-based heritability was determined using GCTA as well. The sample that we used was collected from the apical portion of the cecum and was analyzed using liquid chromatography mass spectrophotometry approach. Here you have an overall analysis for liquid chromatography, mass spectrophotometry, and samples are going to be gradually released from a HPLC column, and this is going to do a characterization based on retention time for each molecule, and each molecule is next exposed to a mass spectrophotometry analysis, which is going to give you a characterization based on the whole spectrum of each molecule, providing MZ values. Those raw files were analyzed with MZ-MINE, performing mass spectrophotometry data processing, and Series 4 was used for the metabolite identification. I'm going to show you 3 different items related to our results, including associated regions for cecum metabolome abundance, estimates for SNP-based heritability, and finally, potential mechanisms driving metabolite abundance in cecum. For the first item, we included a total of 4,861 metabolites in this analysis, and we retained only metabolites that were present in at least 50% of the samples. After the annotation with series, we were able to identify 66.8% of those metabolites, and 63% of those were classified as terpenes. Out of the 0.8 thousand metabolites, 777 showed association with at least one marker. This plot is EQTL-like representation of our results. You have all the associations that we identified in this specific project. The x-axis is showing you chromosomic region that is being analyzed, and the y-axis is showing you hierarchical clustering based on metabolite abundance specifically. The pathways that we were able to include in this analysis were 6. alkaloids, amino acids, peptides, carbohydrates, shikamates, and phenylpropanoids, terpenoids, and uncharacterized metabolites. And you also have an estimate of log p-value for those associations. As I said previously, 63% of those associations involve terpenoid, and you can see the overrepresentation of this specific category across the association plot. This analysis was focused on identifying trans bands. They are defined as loci that are able to explain variability across multiple metabolites. The most important one was identified on chromosome 14, showing you this specific trans band. And it was associated with a total of 149 metabolites. So this is specifically the most important region that we identified in this specific project. This zoom plot is showing you the most significantly associated trait for this specific locus, and this is the specific phenotype, total abundance for tetracosa tetraenoic acid. Inside this specific locus, we identified a cluster harboring multiple enzymes previously described as involved in metabolite composition, metabolite transformations. And additionally, we identified a gene involved in metabolite uptake. So from this specific locus, we identify a number of enzymes, UGT, SULT, CSNs, and one specific transport, SLC. SNP-based heritability. This plot is showing you the whole distribution for 4.8 thousand metabolites that were analyzed in this project. Out of those, 1,668 showed significance after first false discovery rate correction. And you have the whole distribution of SNP-based heritability. Some metabolites with low, medium, and high SNP-based heritability. I'm going to talk specifically about this metabolite. This is a representation for the 7-dehydrocholesterol compound 1. This specific compound showed a heritability of 47.9%. And inside those regions, those QTLs that were associated with this specific metabolite, we identified a number of CPYs. Those are enzymes that have been previously described as involved in the metabolomes of cholesterol. And one of the intermediate metabolites that are generated through this pathway is 7-dehydrocholesterol. So from this analysis, we identify a number of enzymes, again, CPYs. And finally, a potential mechanism driving metabolite composition and abundance in cecum. So as we saw previously, we identified a number of enzymes, SULT, UGTs, CPYs, and CES, and a number of transporters, ABCs and SLC. Talking about the transporters, I'm showing you here the hepatocyte portal vein axis, and you can see that a number of metabolite transporters are located inside the hepatocyte. Some of those SLC and ABCs have been described as able to uptake metabolites from the portal blood to the interior of the hepatocyte. And additionally, some ABCs are involved in secreting metabolites from the hepatocyte to the bile duct in this case. So conclusion, some of the candidate genes more frequently identified inside QTLs associated with metabolic composition in cecum are SLCs, ABCs, GPRs, CYPs, CES, UGTs, and SULTs. And those genes can be classified into different pathways, metabolite transporters and compound metabolomes. Acknowledgements, thanks for your attention, and I'm open for any question.